plankton to the largest phytoplankton, you see something from a size of salmon to the size of the island of Manhattan, no? Is it dead?
Nice work.
All right. So it doesn't matter. So it's really important to understand the scales, okay? This is not like, you know, I'm the smallest one and then somebody's a little bit bigger, and the scales are gigantic, and therefore the physical laws that apply to the areas around them are different. Therefore, availability of the nutrients that they rely on are going to be different. The availability, the way that they interact with different organisms is going to be different. The way that zooplankton is going to find them is going to be different. So in theory, we're talking about completely, you know, this size is not just size, it's just like really defines the way that they're living, you know. We're going really from the ant to the elephant and then to like a really, really, really big elephant. So this is a really important concept to remember, that although, again, high genetic diversity, high diversity in size, high diversity in morphology. Emmanuel is going to give you a little bit more of a talk on particles and how we measure them, how does this translate to the field of optics. But once again, always keep an image that is like, you know, picos are the people, then you have a gigantic whale, and then microphytoplankton is the state of liberty. So just try to think about the physics that applies to these three different things. How does one measure size? This is a paper that Emmanuel was part of that just got published, and it's a really great review, and it gives you a great idea on ways that you can measure phytoplankton, plankton communities, not just by size, but also imaging. I'm going to talk about that later. But this gives you an idea of all different planktonic organisms that we can measure. So starting from the viruses, there are plankton in the ocean, all the way to jellyfish and probably other things that float. So if we would go to that range that I was talking about here, so this is 100 micrometers, no? Here, so we would be here like, so you're 20 micrometers, this is 10, this is 20 here. Then you have 0.2, and where's 200? So 200 would be here. So this is your phytoplankton range that we usually talk about. And you have a large amount of these instruments that are available these days for you to measure, no? Phytoplankton width. You guys are going to be playing with IFCB, which is the imaging flow cytobot, which is able to give you images from which you can define the size, but you're also going to be able to define the taxonomical definition of these species. But these are all different instruments that are available for you these days to measure phytoplankton size diversity. Stop me if I'm mumbling, please. Okay, so this is what I'm going to talk about, size, phytoplankton as particle. I was focusing mostly on size because, once again, we're talking about optics here. Size is really important, as you're going to hear a little bit more. So, taxa. Images I stole from this website called diatom.org, which is a great source for very, very sexy images. But plus, you have quite a lot of knowledge that is on different scales in your availability, you know, capability level. So, taxa, this is a mess, no? Because traditionally, you know, when long time ago, I used to be a phytoplankton taxonomist when I graduated, and I come from Croatia, and this kind of falls on the Vienna School of Taxonomy. So what you would do, you would take a sample of phytoplankton, sit on your microscope for 2 hours, microscope all these, get them all the way to the species, and you're awesome now. And we have a gigantic data set of that. We have like almost 300 years for sure, of these phytoplankton that have been analyzed under the light microscopy, and we have the whole taxonomy that was pretty much based on morphology. But then the new techniques start coming in. We start getting first electronic microscopy, and that allows us to see, you know, on much finer scale, the structure inside, and allow us to figure out that these are actually different species. So we got a whole new slurry of species. Then the molecular techniques start coming in. You know, first genetics, then genomics, then metagenomics, then proteomics, and so on. And with each one of these, we're getting a different view of this taxonomical diversity within phytoplankton that is diverse to start with. No? So definition of phytoplankton, taxonomical definition of phytoplankton is really highly dependent on this concept of taxa that was evolving. And what I prefer to do is just really talk about diatoms or coccolithophores instead that are really maybe not a true taxonomical groups, but everybody understands what's talking about. Because sometimes if you talk about order, they can switch to genera. It keeps on jumping up and down. So I'm trying always to find a medium spot that actually helps everybody understand what I'm talking about, but also I can intercalibrate myself with microscopy-based taxonomy and genetic-based taxonomy. So there's 2 papers that are really awesome to check out. This is a paper that was published in 2004 by Johnson, but Martini, I know him, and it really goes into details about different methods that you can use to establish taxonomical biodiversity of the phytoplankton. And it's just an annual review. And this is the paper that I mentioned before, which is, again, overview of a different, just about quantitative observations of a planktonic ecosystems. What are different methodologies that we should currently use, and where should the field go in order to establish good views and good understanding of the diversity of the phytoplankton. So that, as I said previously, this modern system model classification is 20, 30 years old, depends, the level. And this can be DNA-based, it can be RNA-based, it can be protein-based, it can be additional altistructure electronic microscopy, and so on. And if you want to read about a little bit more about it, Dave Caron, there's a review, there's a link here, and gives you this whole overview how you can use all these different omics to define taxonomy. But usually we refer back to this classical system, which is, again, based on morphological characteristics. They're developed purely from imaging, started from light microscopy, evolved to electron microscopy, and even more recently so, we started using some dyes to differentiate things. So, using genetics, this would be your tree of life. So that's the same image that I showed you before, but this is like encompassing everything. And if you look at this tree of life, you have bacteria. There's no archaea here. Okay, there's no archaea. Oh, there is archaea. You have bacteria here, you have archaea, and then you have eukaryotes. So eukaryotes are the creatures with the real cells, with the real cellable, real organelles within a cell. This is where we are. This is where majority of the algae is. This is your algae, phytoplankton, some phytoplankton here, no phytoplankton here, and then you're going back, phytoplankton and phytoplankton here, no? So they're present everywhere. If you're looking for us, just for the reference sake, where are we? I saw a monkey yesterday. Yeah, that's where we are. All the animals of this here little line. So it's highly diverse, and phytoplankton is everywhere. No, so this is tree of life. I think just using, I'm really not sure what they used to do this one, but then we can like start dwindling down to the smaller groups. So here we're just looking at the phytoplankton. They're eukaryotes. They mean that they're true cells. No, and then here you can see. So this is like, for example, there's diatoms here, these are dinoflagellates here, there's red algae, green algae, and so on. So you can use different techniques to dwell even deeper and deeper and deeper, so then you can do the same distribution, same diversity, even within a certain species. So this is Perchlorococcus, it's a cyanobacteria. And here they use 2 different techniques to look at the diversity within single taxa of them. And by using the RNA or the whole genome, they found so many different types of the Perchlorococcus. So from your perspective, you can go and do this whole thing, but what is it going to bring to the table? The only thing that you care about is this diversity anything to do with the optical signal, and is it important for you to understand this whole diversity from the questions that you're asking to understand ecosystem or to understand carbon cycle? But important thing it is that you can use these genetic or omics techniques to really dwell deep from the diversity of life to diversity of group or to diversity within certain species or taxa. And sometimes they're going to give different answers. But cool stuff that you can do with these things is that you can, like, a very easy and very nice way, look at the distribution and diversity across different oceanic realms. No? So this is the data set from Tara, and if you ever want to play with DNA or like just omics-based data, that's a great, very available data set. And what they did here, they used this as metagenomics data, if I remember. And Manuel is not listening, so it doesn't matter. It's metagenomics data. And where they collected all these samples all around the world, and they were not looking only at phytoplankton. They looked at the whole plankton structure, and everything that is with greenish up here is a phytoplankton. You can start then seeing the diversity of this phytoplankton, similarity of the planktonic communities around the world. And just like, it's a really beautiful way to show that, like we think about the ocean as one thing, but actually it's full of these different ecosystems and these different like forests and jungles and maybe deserts and stuff. You can like, one of the coolest things that I always liked is, I don't know, have you ever looked at the circulation of the ocean, but what happens here, that you get these like teeny tiny rings that start spinning here. whee, and they go this way, no? So what Tara did, they followed these rings that form in the Indian Ocean and can transfer inside of the Pacific. And what they found, that a really large amount of genetic material from here gets injected through these eddies up here. And they use genetics to do that. They use, OTU-based, or this is metagenomics, systems to actually follow the trace of how much of the genetic material of the phytoplankton, plankton in general, is getting transferred from Indian Ocean to here. So you can do these cool studies and start looking, in a much nicer way to the diversity of the ecosystem and answering questions that you're interested in. But the problems kind of starts coming is when you start relating that to the traditional microscopy. So what I'm seeing a big problem these days, and I'm seeing more and more people doing so, that we have, again, 300 years or 350 years of this traditional microscopy. And then we have this budding, beautiful genetic techniques, omics techniques, and they're kind of like living in this separate world. So we have this historical data that if you don't do these two things parallel, you're going to have disparate data sets. So you can't do this historical analysis of the things. But luckily, and that was mostly because this microscopy on light microscope got me out of that field. It's 2 hours per sample. So development of these new automated imaging techniques are actually allowing for this disparity of these two data sets to disappear. So we have these automated imaging techniques. They're like taking samples for you, taking photos for you of all these species. We have, AI or different neural networks actually helping you analyze this data and giving you the taxonomic composition. If you took the DNA, at the same time, you can start making these translators, no? So we're actually entering in a world where we can start combining these data sets. So once again, this is a classification based on morphological characteristics. Started with light microscopy, but now we're using this imaging, and also scattering techniques. Electron microscopy allows us to see the ultra-structure of the things, and we started using pigmentation to a certain extent. So you can, when you're using microscopy, you can use pigment-specific excitation lights. So, for example, Colin was talking yesterday about chlorophyll. But there's other different pigments that you can use, and you can excite them with the specific lights. They can allow you the distinction between two different groups. You can actually also use some dyes to pull out certain things. For example, heterotrophs or mixotrophs. You can dye them, and you can pull them out under microscopy or any kind of scattering systems that you can use to differentiate different taxa groups. So when it comes to this taxonomy, this is like, I'm just going to go through a bunch of different groups. So cyanobacteria, teeny tiny creatures, phytoplankton, smallest phytoplankton. No, once again, they're bacteria. What that means that they're not true cells. They're not cells like we are, not true cells there. They don't have any organelles. Eukaryotic cells have a nucleus. They have a bunch of this mitochondria. They have chloroplasts. They have different things. Bacteria doesn't have that. That's a bowl with a bunch of stuff floating inside, no? And if you're looking for a chlorophyll or favorite pigments, they're going to be kind of like stranded in the membranes of this bacteria. We didn't know anything, or a little bit, about cyanobacteria. The most abundant cyanobacteria, Cetinić and Prochlorococcus, were discovered within the last 40 years. Me and Cetinić are the same age. It was discovered in 1977. I was born in 1977. So, we didn't know anything about the most abundant phytoplankton until 1977. And again, once again, development of technology allowed us that. Prochlorococcus was discovered in 1986. And if you read about it, the amount of primary production and the abundance of these creatures in the ocean they're the dominant thing everywhere, no? You have Trichodesmium, which are like things that I really adore because they look like puffs and they look like my hair and they're really awesome. And you're going to find them in the tropical areas. And what is specific for a majority of these cyanobacteria, that they have a capability of nitrogen fixation. So probably all of you have at least one plant in your life, or you have killed one plant in your life. Everybody interacted with the plant, and what plant needs is sunshine, water, some CO2, which you have, but you need some nutrients, you need some fertilizers, no? And that's what the phytoplankton needs as well. Sometimes this doesn't come so often. Like if you're in the coast, you're good because the rivers bring stuff. There's upwelling, there's yum nitrogen and other things. But when you're in the open ocean, things kind of start getting scarce. So thankfully, these are bacteria, so they developed a system to get that nitrate, that nitrogen from the air and actually fix it in their body so they can use it for the, whatever the needs are in a cellular system. So they're really, really highly specific. You're going to find them all around the oceans, especially in tropical areas, and sometimes they form, they tend to be symbionts with some large things. So going back to my, Cetinić one thing, Percolorcoccus, this is just a paper showing a distribution. Again, we didn't know these things existed until 40 years ago, which is amazing, no? So Sin, Pro, Global, this one goes all the way to 50 north. This is something that is probably that's causing majority of the chlorophyll that you see in the open ocean. Really important to remember, they have specific pigments. Some of them, Sin has phycoerythrin that allows us to fluoresce in a certain way, and that's the way that we can kind of catch them and distinguish it from the rest of the cyanobacteria. Trichodesmium already mentioned. It can be puff, it can be just crazy looking thing, and yeah, it's present in a tropical ocean. If you have ever been on the ocean anywhere in tropics, so Sargasso Sea or Red Sea or anything like that, you're going to see slicks of these things. So although these are cyanobacteria, like maybe around 2 micron or less in size, they start forming these colonies that float on the surface, and they make these surface slicks that are really, really visible. So from one perspective, if you study them, you can be very happy, but if you sailed into a pile of trichodesmium and you're trying to do carbon, it's really, really hard. And we can see them from space. So this is off, God knows where, but probably somewhere like Southern Ocean or like off Australia. You can see this. Does anybody know what this is? Thank you. Well, I was correct. I was at least part of the hemisphere stuff. Yeah, so this is this trichodesmium bloom, and you can see them, usually around Christmas time. I mean, everybody's Christmas time, but that's like, you can see them around Christmas time around Australia, This is a gigantic group that I like a lot because, they're pretty awesome. I can't pronounce this in English, just read it. This group contains 2 big subgroups, coccolithophores that I mentioned. They're really awesome, and we like them in optics because they have this calcium carbonate shelf. So take this chalk and grate it and put it in a glass of water. What are you going to get? Lots of chalk in the water, but how is it going to look like? Like milkish, Therefore, we love it because we can see it from space. When they bloom in large amounts, they're going to start changing the water to milkish turquoise. Awesome. We have PFT now. We can recognize phytoplankton grew from space. Not all of them have a capability of forming these calcium carbonate shells, but this is generally thought of the characteristic of all the coccolithophores. They're really important in geological times. Most of the structural geology or oil geology is using them to trace and find the old depositions, because based on the different species that evolved, in different times of different geological time, they can kind of go and pin down where the oil is. They're very sensitive to sea surface temperatures, and that's why they use it as a tool, paleontology. But there's lots of work recently has been done on using them as a tracer for the climate change and changes in the temperature conditions. They're kind of a little bit of a princess of the phytoplankton world. They really like lots of light and specific temperature range. I'm over-exaggerating. Some of them you're going to find everywhere, but majority of them you're going to find them blooming usually around solstice. So if you pull out any kind of like MODIS image or anything now from the northern hemisphere, you're going to see a bunch of these patches of the coccolithophore bloom because solstice is coming up. They really like lots of light, and they're very, very sensitive to temperature because the calcium carbonate shells, you can assume right away they're sensitive to the pH, but there's been many, many studies showing a bunch of different things. I'm not going to say, I'm just going to say sensitivity to pH. They're extremely important for the carbon export. On the other side, one other player in this group is Phaeocystis, which is not as sexy as this one. Honest to God, the thing makes not this big, but big blobs of a bunch of teeny tiny unicellular cells that's sitting in this goo. And it doesn't look sexy at all, especially when you have these blooms. It forms this disgusting film. So if you have ever been in England or something like that, or just on a beach when there's like this like disgusting looking film, that's your Phaeocystis bloom, And it's a really strong producer, this DMSP, which is a climate gas. It has the capability of forming clouds and similar. But they're also really important from the carbon perspective because they can draw down quite a lot of carbon because they're just gigantic pile of goo that sinks in the ocean. Problem here is that they're really big, so usually we cannot use optical tools and CT optical tools to kind of characterize them really well. But they're an important group to remember. Once again, some cheese grate some of your chalk and put it in the ocean. This is what you get. I think that's the same image that Colin was showing. Yes, this gigantic, gorgeous bloom albicoccolithophora. And that's pretty much the first species that we were able to recognize from space, just using basic technology that we have, just because of the fact that that is forming these blooms. And I know where this is because it says Norway. Dinos have a bad rap, but they're beautiful. So some of the images, and then there's many, many more. They're creatures that, again, we always thought that they're solely autotrophs. As we're learning more and more about them, we start knowing that the majority of them are heterotrophs, meaning that they can do photosynthesis, therefore, like, draw down carbon dioxide, produce sugars, and produce oxygen, but they can also kind of go and start eating other things and finding other sources of carbon and other elements that they need to survive. They have a flagella. And opposite to flagella means a little tail. Usually they have two tails. And opposite to everybody's belief, they didn't do this. They really do this. They kind of follow their tail. So they are movable. That we thought about them, they're drifters. They're movable to a certain extent. I mean, if there's a big ocean current, it's still going to take them away. But sometimes, for some of them, you can see the swarming behavior. So for some red tide blooms, or like Noctiluca, or even I think like Lodinium also has this capability. You're going to see them, like especially in the areas with the currents, you're going to see the swarming behavior. But again, this is in constraint of their physical environment. If there's a big current, they're going to go bye-bye anyways, no? You're going to find them mostly in coastal and warm waters, therefore like red tide blooms. These guys are the ones that you find in coral reefs. They don't look this way, no? But you know that in coral reefs, you have the symbiotic algae. They live in coral reefs, give them oxygen. Algae, the coral reefs give them protection. Everybody's happy. This is the guys, this is the group that is going to be in there. Once again, the changes in temperature and changes in light availability can make these guys die make dinoflagellates die within the coral reefs, which ultimately leads to the death of the coral reefs, among other parameters that kills coral reefs these days. Again, some of them are toxic, but they really got bad rep because of that stuff. Quite a lot of them are toxic, and we can talk about toxicity some other time, but that's kind of not important. This is the photo that I took during my PhD, California coast. This is a bloom of one of these red tide species. I mean, ocean in California usually looks like grayish blue. This is how I look. This is Lingulodinium, one of the prime suspects there. This species is not toxic itself, but just by looking at the color, you can see there's lots of them. So in coastal areas, the species can be harmful, not just because of their toxicity, but when you have lots of them blooming, eventually they're going to run out of the yum-yums, of the nutrients, fertilizers, they're going to die. So once they die, they sink. And what happens then? Does anybody have compost at home? Or you forgot some organics? Yeah, it starts stinking pretty bad because the bacteria comes and starts eating that organic. And bacteria is going to use up all the oxygen. In the areas with really bad circulation, you're going to have a decrease of oxygen in the bottom area or complete disappearance of oxygen. And then all the fishies, all the lobsters, all the things that live in the bottom of the sea die. So that's the harmful, so it can be harmful because it's toxic, but it can also be harmful because in disentrainments or coastal areas where you have shallow water column and low circulation, they can cause a lack of oxygen and then death. And diatoms, okay, these are the queens, the most pretty ones. I mean, so you're going to see, if you go through the slide, it says Sardet here. Sardet? Am I correct? Sardet? Okay. So Christian Sardet was also associated with Atara, and he has this like amazing microscope, kind of dissecting, kind of real microscope. And while in Tara, he was taking these amazing, amazing images. And while I was in Tara, I was trying to take an amazing image, but it just sucked. I could not. However, he took this image. And I mean, this just like looks like pure gold. And if Google his name, you're going to find many, many different videos and everything that you're going to find really inspiring. But these are two beautiful images of the dinoflagellates. Dinoflagellates, once again, single-cell organisms, but they very often form colonies. They're diatoms. Single-cell organisms but very often form colonies because that helps them to survive, take nutrients, stay afloat, or protect themselves from the predators. If you look like this, if you find something like this in your zooplankton, probably you're not going to go after it. I mean, kind of looks scary, you know? But we find them to be really, really important because they're the harbingers, like they're the first ones that show up in spring. Now, so if you think about spring bloom, you go in the forest, you find like certain species there that show the spring. This is the spring in the ocean. So North Atlantic bloom, the classical one, the most important one that everybody studied their phytoplankton ecology on happens in North Atlantic, like the green stuff and blue stuff up there. That's thanks to these guys. So what happens, they bloom, they feed the zooplankton feeds the fishies, then Gulf of Maine fishermen, Iceland fishermen, Norway fishermen, everybody has cod, you guys have cod, herrings, whatever you want to eat. That's really cool. If they don't get eaten up, they die. Once they die, because they're big and they can produce polysaccharides, they get sticky to each other, form marine snow, and sink really fast out of the water column, taking all the carbon out. They're really superbly important, abundant, and beautiful. There's two different forms, pennant and centric. I'm not going to go into these details. Some toxic, some of them are toxic. The most popular one is Cetinić. I'm really giving you here the stories of things. Cetinić. So, did anybody watch ever a Hitchcock movie? I mean, you know all the stories, so don't smile there. Santa Cruz, boy. Hitchcock? Okay, yeah, last. Okay, birds. You guys heard about movie Birds, you know? There's crazy birds going and attacking Tippi Hedren. You know, I was like, eh. So the story goes that Hitchcock, that guy, spent his summers in California in a city called Capitola. Thank you. And supposedly he was like, taking a stroll. I can't see him taking a stroll, but standing there, and he would start seeing birds acting weird, kind of like smashing into things and everything. And supposedly that's we got an inspiration for making the movie Birds. So we know later, and it's suggested because we don't have the analysis of those birds, that actually happened because of the Cetinić, because of the diatom blooms. These diatoms form a toxin that makes birds and marine mammals like act loopy. So while I was in California, there was actually quite a lot of like seabirds just like slamming into cars and vehicles and by accident and people, just because the toxin messes up their brain. Without going into definition, I mean, you can flush it out, but because this toxin accumulates in sardines, birds come and eat sardines, mammals come and eat sardines, the toxin accumulates in them, doesn't affect fish, affects mammals and birds, and then they kind of go loopy and probably die. So this is why this toxin, domoic acid, is usually of a big concern. It doesn't affect only mammals, it affects us as well. So one of the first, I think, known deaths of a phytoplankton happened in Prince George Island in Canada. It was actually it was a Cetinić diatom bloom where somebody dropped dead from eating oysters for domoic acid. So once again, how can you use optics to help the humanity? You can actually develop algorithms to follow specific types of phytoplankton and use it as a warning signal for the aquaculture community, especially here in this area. That's what they do here. So they stop harvesting the mussels and oysters who are filter feeders while that bloom is happening, and after a week or two, they flush it out of their system and you can start eating them again. Okay, now we're going to take a break because I've been talking a lot. Okay, some of you might have taken this quiz, no? So if you have not taken this quiz, I want everybody to take this quiz. So if you have a cell phone, you can just scan the code here, or just go Google PACE. NASA is going to take you there, and then and there's a phyto quiz, and I want you to take it. So this is the quiz that we made for the PACE mission, which is that up-and-coming fire-perspective mission is going to change the way that you see the world and cook you espresso coffees on the side too, and we kind of tried to find a cute way, for millennials, because everybody likes the clickbait, to kind of drag them to the website. And once you start doing, are you doing it?
No, I did it already.
Do it again, because you're not getting it. So everybody's gonna get the tattoo. I have a phytoplankton tattoo. So whatever species you get. So the way we made it is we start thinking about all these things that I talked about. Okay, I'm gonna be talking about a little bit more about pigments, but you're gonna have a question is like, when you're in a party, when you're in a pool, do you like to swim? Do you like to sing? Do you like swim like a champ? And think about dinoflagellates. They have capability of swimming. Do you sink? Diatoms sink, no? Do you just float around? Like Trichodesmium, for example, floats around. And that allows us to figure out which one of those species are. The next question is I think when you go on holidays, where do you like to go? So we talk about cosmopolitan species like, Synechococcus superchlorococcus that is present everywhere. You like tropical weather, you're probably going to be Trichodesmium. If you like cold water, you're going to become a diatom. Then your next question, I think, is do you like to hang out and party with people? Colonial species, or you just want to get out, single cell organisms, they can swim. things like that. And the last question, which color do you like? And that's associated with the pigment composition. And it's really cool to see how people start doing this stuff. And it's really, we kind of recommend the teachers to kind of group their students into those different groups that kind of define the traits. And I know kind of the probability of what you're going to be. We do have a probability matrix, but sometimes I'm surprised. So, I mean, it's only 4 questions. Let's see. Are you Alexandrium? That's cool. I'm a trichodesmium, just so everybody knows. I'm like a tropical species with bad hair. I took this before. I got the same thing. What do you have? Dinophysis. Okay, so we have 2 dinoflagellates. Another dinophysis. Dinophysis? Okay, there's lots of dinophysis. Whoa, dude, we're having a bloom here. I think there's a space, yeah. Just Google PACE and NASA.
PACE.
NASA. Yeah, so go on our website, and there's going to be a quiz on the first page. I asked them to push it out.
Take our quiz?
Yeah, okay. Take a quiz, yeah. So if you cannot find it, just Google PACE, NASA, and the front page, it says like big button like this, take our quiz. So we have, 4 dinophysis, 1 Alexandrium.
I got a
Cetinić, woo! Cetinić, Sin? Protoperidinium, dinoflagellate again. Do we have a diatom here? Alexandrium? Yeah. You're like, we were with Emmanuel. You? Dinophysis? Not a dinophysis. You? Cetinić? Really? Guys, the biological diversity sucks. Come on. What, do we have any? Alexandrium, do we have anything? Microcystis. Now we're talking. That's a rare one. Marionecta rubra. So Marionecta rubra is actually a ciliate. It's a zooplankton that has a symbiotic algae in it, no? But we kind of consider it to be a photosynthetic ciliate. So it's one of those weird ones that can do left and right. That's a pretty good one. That's a really rare one. I wonder what took you there. It's the red color. It's the red color and there's something else. I'm trying to think how we structured the test. Anybody, anything cool? More dinos? What is it? Read the name. Rhesusolinia. We have a diatom. Yay! He can have a symbiotic cyanobacteria. He can float and make big mats, and he's very important for the carbon export as well. Anybody else? Anything exotic? No? Okay, try to remember your phytoplankton. Now have a little tattoos, okay? So I'm going to get you tattoos, and you can proudly wear them. They're going to probably wash out or probably not really stay for a long time, but Now, and try to, think of, it leads you to the website that tells you a little bit about it. And then if you read about it later on, you're going to figure out why you got that stuff. And usually it turns out it really fits with people's traits. So what did you get?
Alexandrium.
Alexandrium, another one? Dude, guys, you're toxic. Dinophysis and Alexandrium are both toxic. That's, I don't know. I don't know any more recent songs. It's the only thing that I wouldn't say toxic is Britney Spears and Toxic in my head. Really bad. Okay, awesome. Okay, this was a break, so everybody can giggle, get some oxygen into you. Let's go to a little bit more through the chemistry. So talk about pigments. So this is a fantastic image. And it's really hard to get this stuff. So these are a bunch of different cultures of algae. So cool stuff that we can take these phytoplankton and we can culture them. We can grow them as plants, You give them some ocean, you give them some light, you CO2 they have, put some yum-yums, put some nutrients, and you can start growing them. Not all of them, but majority of them. So what you're seeing here is a bunch of different ones. This is a Perchlorococcus, who turned out to be Synechococcus. She was Synechococcus. So this is a Synechococcus. You see how beautiful pink it is? This is a Millania huxleyi. So it looks yellowish here just because it hasn't started making its calcium carbonate shells. Millania does that when it's kind of a little bit freaked out with phosphate, so they haven't still freaked it out a bit here. This is a calcificata, probably some another calcium carbonate. This is a diatom. See how nice and brown it is. And this is Tetracelamis. Let's call it the green algae just for the sake of the conversation. You can see all this diversity of colors. And now you start thinking, yeah, of course I can see them from space. No, duh. Look at how colorful and different that is. So these colors actually are mostly, we can contribute to the pigments, And pigment diversity within phytoplankton is large. Think about the pigment diversity on land, all these different colors of the flowers. You have that within phytoplankton cells. And this pigment diversity, we can structure these pigments as light harvesting. So that the pigments, they work in together with the chlorophyll to help harvest the light, catch that light, transfer the light and stuff. I mean, the photophysiology is a little bit more complex than just having a chlorophyll, but we're not going to go into that. Then you have photoprotective pigments. Well, a little bit of sun to get you nice and tan is awesome, but a little bit too much sun can kind of destroy you and get you cancer. The same things happen with the cell. A little bit too much UV light can start, destroying the cell and do bad stuff, especially damage to the DNA, So that's why they have these photoprotective pigments. These are the pigments that kind of come in place to protect the phytoplankton cell from bad effects of the sunlight. So we have chlorophylls. Now they're green. That's easy. Colin talked about yesterday a lot, and I'm going to touch a little bit. And that's going to be your, that's the pigment. Everything is green because of the chlorophylls. There's tons of different chlorophylls. Chlorophyll A is the most abundant, but then you have chlorophyll B, chlorophyll C, chlorophyll D, you have divinyl chlorophyll, and different types of chlorophyll are actually associated with different types of these taxonomic groups. It's just chlorophylls that evolved in a different way. So there's a bacterial chlorophyll that actually exobiologists are now using to try to develop optical ways to see it in these usually extreme lakes, and try to think about using that optical signal for these bacterial chlorophylls to look for the life on the other planets, the ocean worlds, for example. You have carotenoids. These are brown and orangish. Carotenoids, very easy to carrot, has lots of those. You have beta-carotene, that's the carrot one. That one is very abundant in a phytoplankton world. Then you have xanthophiles, Peridinin, that's your dinoflagellate friend, allotaxin, and zeaxanthin are happening in other species as well. And then you have phycobilins. Phycobilins are the cool ones. These are the ones that you find inside the bacteria. And they can be blue-green or reddish. So phycocyanin and phycoerythrin, both of them fluorescent, and we can find them in the cyanobacteria. These are gigantic groups. This is kind of like your gigantic definition of pigments. Each of these have a little teeny tiny subgroup. And highly depending on the technique that you use to detect these pigments, that's going to allow you to differentiate more and more. However fine your technique is, it's going to allow you to differentiate more and more stuff. So your Royal Highness chlorophyll, Colin touched upon it. It's really important to remember. There's a string up here, magnesium in the middle, and then there's a tail. This tail allows it to sit inside of the membrane complex, and you're going to see a little bit more. This is my Christmas tree. When chlorophyll gives you a paycheck, you put it on your Christmas tree, That's pretty much what pays my life. I work for NASA. But so one thing that was really kind of for me, it was always chlorophyll, and kind of I had this vision of where the chlorophyll is. So I'm just going to show you a couple images to understand how this chlorophyll looks. Chlorophyll is really important, again, because it captures the light and starts pushing these electrons down into the magical things, the little goblins of the cell. But it doesn't kind of sit just like in a membrane. You see these little green little thingies? That's your chlorophyll molecules. The yellow things are little photoprotective, photo-helping pigments. This is probably some kind of beta-carotene or something like that. All these wiggly things, these are different proteins. So all these chlorophylls are sitting in this gigantic molecule, build a bunch of different proteins, all the other helper pigments, and this is your photosystem 2. Okay, so now you take this photosystem 2 and you put it inside of the membrane, Then you have all this photosystem 1, you have a bunch of different inclusions inside of the membrane to make the magic happen, So once the light hits that chlorophyll, it has to travel, this electron has to travel through all these magical things to make something happen in a cell. So just think about it, it's not just that tail sitting inside of the membrane. Tail keeps it connected to the protein sitting inside. There's a bunch of these complexes in the cell that make the magic happen. And it's completely outside of the scope of this or this class to explain you everything. But if you're interested in this, and it's really awesome, I can definitely direct you to techniques. As I said previously, you have eukaryotic cells like us, like plants, like majority of the phytoplankton. They're true cells. And they have organelles. So this is your chloroplast here. And inside you have these thylakoid stacks, in membranes of which little phytoplankton lives. And in prokaryotic cells, there's no organelles, there's no chloroplast. So everything is kind of floating around. So your DNA is just floating around, it's not a nucleus. And then chlorophyll, these chlorophyll complexes are going to be living inside of these cells here. instead of the cell walls here, entraining this membrane and actually doing their stuff here. And it's one thing that you're going to see that these two things look very similar. And it is because the chloroplast actually evolved from one of these bacteria that was by accident either ingested or symbiotic process within a larger cell, and that's how the prokaryotes happen. So carotenoids, I think I'll mention them or not, getting beta-carotene, carrot, remember carrot, orange stuff. They're really important part of that photocomplex. They help chlorophyll deal with that light and transfer light. Xanthophiles, these are the photoprotective pigments. These are the ones that start appearing in a cell and kick in a cell to help deal with the extra, with the sun damage and extra light that is hitting, you know? I'm not going to go into details there. There's many other pigments. Why do we like pigments in optics? Because they have their absorption spectra. Colin showed you a couple absorptions yesterday. There's so many, there's a whole field of this that is really devoted to the absorption of pigments, and depending where are you measuring these pigments in vivo, so it means inside of the cell, or in vitro extracted from the cell, which kind of liquid do you use? You use like acetone and methane and so on. Their absorption, especially their excitation and emission wavelength, going to look different. But this is your absorption here. These are your typical pigments that we use. So if you would collect a sample to send for the pigment analysis, pretty much these are the pigments that you're going to get. How do we do that? The way that we measure pigments is using high-performance liquid chromatography. And you're gonna see us using this word HPLC, 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 HPLC. So HPLC is a technique that is used for analysis of many different things. It's actually owing to the fact that all these different pigments have a different mass, different size, different complexity, and they travel with a different speed through column. As they're dripping out through the column, they're measuring their absorption capabilities, and in comparison to the pigment standard, they can differentiate different pigments. So once again, chlorophyll C, pyridinum, there's your dinoflagellate also, lutein. So these are all different pigments that are popping up out of that column, and then they measure the absorption. That's how to do it. If you're crazy enough, go and measure HPLC by yourself. I would recommend that only if you come from one of 5 labs in the world. Do it by yourself. If you're a normal person, just collect, filter really nicely. We'll teach you how and send it for analysis. Your life is going to be a much better place. Pigments are awesome because A, we can see them, but they also relate. We can use do some taxonomical stuff with them. Why? Because as I said it before, when I showed the picture of that chloroplast and where the chlorophyll is in, and the picture of the cell, of the prokaryote, they kind of look the same. And that's true because long time ago, there was some kind of like a cell, and then another little cell came and kind of started doing some symbiotic relationship with it. And that's how the prokaryotic cells actually happen. And that's how the chloroplasts happen. And it happened several times in history. We're talking about evolutionary history.